Hey guys, Dr. Erica Wood here. We're going to talk about mitosis and hereditary. A different view of my office today is because my back is out. I'm going to share my screen and let's go. Away we go. Okay. So first week we had a very brief overview of what cellular biology is all about. We're going to dive back into that. Feel free to review your notes from week 1. Prior to coming to this lecture. It's good review for the final, but it's also good to have some knowledge that you can back yourself with. So as far as mitosis and meiosis go, let me just close this part out here. I was all set up and ready to go, and then I wasn't. Okay. So as far as review, so remember that everything that is hereditary is in our DNA, and DNA it's housed in our nucleus, and the nucleus has 3 main structures that we need to know about: a nuclear envelope, a nucleoli, which is like the center house of it all, and chromatin. Chromatin is where our DNA is collected. Nucleolus is where RNA is collected, and nuclear envelope is similar to like the plasma membrane. And why we need to know about that is because as part of the cell cycle, these areas play a huge role in that. Okay, so cell cycle is the series of changes a cell undergoes from the time it's formed until it reproduces. There's 2 major parts of the cell. There's interphase. An interphase is actually the longest aspect of the cell cycle, and its job is to help the cell grow and carry on its activities. And then the shorter phase is the actual cell division, which is mitosis. Mitotic phase, mitosis, which means that the cell divides into 2 different cells. And with mitosis, you're going to have the same amount of chromosomal DNA as far as pairs of chromosomes as you did from the beginning. Okay, so it's called, in humans, we are what's called 2N or diploid organisms. So after mitosis, you're going to still have a diploid cell, and that means 46 chromosomes. So an interphase, it's basically from cell formation to cell division. And then during interphase, the chromatin, the area that our DNA is housed, this nuclear material is condensed in a chromatin state. And then during interphase, we have different phases in which the process of DNA replication occurs. So the idea for mitosis is not just to split the cells with no information. We want to replicate the information that we want to clone and keep going and moving forward. So the subphases that we need to know about are what's called gap 1, the synthetic phase, and gap 2. So gap 1 is the part of the interphase where there's a lot of growth and metabolism. When cells that permanently stop dividing, they are said to be in the G0 phase. So just that's important. G0 means that they're no longer dividing. They are what they are, and until they die. Then we have the synthetic phase, which makes sense. Synthesis means to make or to make. And that's where all the DNA replication occurs. And then there's G2, which is gap 2, which is preparation for the cell division to occur. So right here, as far as when we talk about, when we think about things like cancer or mutations, we have lots of checks and balance systems and interphase. Basically, what's happening is that it gets almost like a scan. So right before it's about to, while it's growing, there's a scan point, kind of like when if you're walking into any kind of office setting where you have to get your temperature taken. We want to make sure your temperature is nice and right before you come into our office, because we don't want you to spread the Rona. Okay. So first part of that phase is G1, so that's a lot of growth. And then at 1, there's a checkpoint that if that G1 does not pass go, then it stops and that cell dies. But as long as everything checks out right and there's no mutations or concerns, then that checkpoint is established and it moves forward to the S part, or the S phase, which is where the growth and DNA synthesis occur. Then we have the gap 2, once again, which is growth and the final preparation for division. And then we have another checkpoint here at G2 between gap 2 and mitosis phase. Because before we replicate and make these cells go, become more, we want to make sure there's no issues. Now, the issues with cancer is that how cancer comes about is that sometimes in G1 or sometimes in G2, the mutations occur and it kind of tricks the checkpoint system, and the body doesn't recognize it and keeps going. And there's certain genes or even certain proteins that are exhibited that kind of stop this from occurring. Typically, you'll hear about something called RAC gene, and RAC gene is important with this aspect. And then we have the mitosis phase. So you see here, large part of the cellular mitosis is in interphase, and the tiny bit here, about 1/4, is in mitosis, or the mitotic phase. Okay. So mitosis, we have two different distinct aspects, where the actual mitosis itself was the duplication, and then we have cytokinesis. And most cells need to replicate to continuously grow and repair. So if our skeletal muscles, our heart cells, and our nerve cells don't divide and conquer, then if we damage those, they don't divide and conquer the proper way, then they are damaged and we just get scar tissue. What happens with scar tissue in the cardiac or in the nervous tissue? Scar tissue in the brain can lead to stroke or hyperperfusion. Cardiac tissue, that's an MI, that's myocardial infusion, and same thing with skeletal muscle. Skeletal muscle can atrophy and die. And I apologize, you're going to hear me say death, death, death a lot because we're talking about cell division here. As far as what controls cell division, we have to, those little proteins and checkpoints are really important, because if that control is not there, then we might have division of cells that go like crazy, and that is also cancer. Cancer is normally that we have an overgrowth of cells that we don't want that have mutations. Okay, so there's different phases of the cell division. We're going to go to each of those in depth. So each phase has a different distinct characteristic. And if we were looking at it under a microscope, you can actually tell what phase of mitosis a cell is in. And this is to ensure, why we have these different phases, is to ensure that we're getting a full copy of replicated DNA. Hopefully no mutations and no duplications or anything like that. So another thing to understand is that DNA is that master blueprint that holds the code for protein synthesis. So if that DNA replication goes awry, our proteins go awry, etc. And DNA also, it's responsible for, remember from week 1, that DNA translates to mRNA then goes to tRNA, lays down the amino acids to make a polypeptide. If that DNA code is wrong, then that polypeptide is wrong. And a segment of DNA that holds the code for only one polypeptide is referred to as a gene. So every single trait we have is a protein that's expressed in our body or not expressed in our body. And then to make sure that this divide and conquer occurs properly and make sure that we aren't, for the most part, not producing cancer cells, we have different processes that occur in the cells. So if the cells are damaged or they become obsolete or not needed, they either go through autophagy, and autophagy is basically the self-eating process of the non-functional organelles and little bits, and it gets degraded by lysosomes. If you remember what lysosomes do, they have a lot of enzymes that degrade. And the unneeded proteins that are left over are marked for destruction by something called ubiquitins. And these ubiquitins also use proteasomes, and things that are highlighted on this slide I want you guys to know. Proteasomes, what their job is to do is to disassemble these ubiquitin tab proteins. So it's like going to Ross right after Rona allowed you to go in. So after it opens up and everything's from winter and it's now spring. Everything gets tagged for clearance because they want that to go fast and get out of there. So your ubiquitins are kind of like your clearance cell. Tag it, get it out, get ready for new. And then there's another different type of thing that's different than the autophagy, which is apoptosis, which is a programmed cell death. So an example is that if something goes awry, then things like infected cells, old cells, cancer cells, they go on self-destruct, meaning that they know that there's something wrong, they should self-destruct. When these mechanisms get overturned, which is common, this is how viruses and cancer cells have figured out how to get around this and duplicate and replicate. And then as far as aspects of development aspects of cells, so all cells of the body contain same DNA, but not all cells are carrying the identical types or carry out the same function. So example, your ovaries or testes have a very different job than your adrenal glands. And that's because the DNA is the same in in every cell, but not all the cells are signaling for the same mechanisms to occur at the same time. And then there's chemical signals that we received in utero as an embryo that help turn specific pathways and developmental pathways that help turn genes on and turn genes off. Fun fact about Dr. Wood is that in undergraduate, when I was a molecular pathology major, I used to do something called transgenic mice. And what we would do is purposely knock out genes to see what they did as far as how that embryo would develop and how that gene influenced that development. Disgusting. I was a vegan. I felt really bad about it because I had to do some not nice things to those mice, but it was important to learn about those things for things like diabetes and cancer treatment. We also, with development, that we find that each type, the way that these signals are carried out, they give each cell a distinctive feature, and that causes cell differentiation. So we all start out as this little stem cell, and some of you guys might have heard about embryonic stem cells, but then through this signaling system, our cells now know what to do and how to become different. There's a couple of ways that cell division can go awry, and I will get into the phases of mitosis in a moment. I want to make sure I touch base on some of these. So cell destruction and can occur, but organs, as we know, are well formed and function before birth, and we need cell division to grow because we don't come out full blown human size. I would pray that no person ever had that happen, unless curious case of Benjamin Button or something. And then also that we go through we know every 120 days our blood cells are renewed. Our liver needs to regenerate. Our brain cells need to kind of keep making formations and pathways. So cell division in adults needs to replace these short-lived cells. And if I accidentally went with the scalpel blade on my wrist, which I have as doing autopsies, it was not fun. We need our body to be able to regenerate. And that's what's important about these cell divisions and how fast it can occur. When something is happening a little too much, too quick, it's called hyperplasia. Hyperplasia is not the size of the cells that are increasing, but the amount of the cells that are increasing. Sometimes we'll see hyperplasia occur right before a cancer is about to occur. And we kind of, it's especially in this example, endometrial hyperplasia means an overgrowth of endometrium. Sometimes that could lead to cancer, and we try to nip it in the tissue before it happens. The opposite can happen, which is atrophy. So you might hear that with the terms of muscles, where there's a decrease in size of the cells, and that results from the loss of or stimulation or use. Now, there is a term that's not on here that you might hear interplayed, and that's hypertrophy. Hypertrophy means that the actual cell size itself is growing. So when we're growing our muscle cells or our cardiac cells, we might have hypertrophy, and that's the actual increase in the cell size. Hyperplasia, number of cells are increased. Okay, well, I need to readjust how I'm sitting here, because yes, my back is out, but now I can't feel my toes. First world problems, I know. Okay, so as we all know, we grow, we get older, we shed the mortal coil, and whatever your views are of how things come about. But we don't really know exactly why we age and why we wear and tear. There's a lot of different theories out there. Some of them are like telomeres, which are little buttons on the end of your chromosomes. We think that every time a cell replicates, those get shorter and shorter and shorter. And when those get shorter, we don't have expansion as we normally do. Wear and tear, so chemicals and free radicals that have a cumulative effect over time cause our cells to age. A lot of this can be considered as, think of sun damage when you're sitting in sun exposure. Lots of free radicals being made there, and even though you had tons of tan, and I know I tan really easily, it's my background, can't help it. I wish I didn't tan so easily within even 5 minutes of being in the sun, neither here nor there. But free radicals that have accumulated over the years can cause things like skin cancer or cell death. Mitochondrial theory of aging is that these free radicals are in the mitochondria. Remember that mitochondria are the powerhouses of our energy, and they diminish energy production. Big thing in naturopathic medicine for longevity health is the idea that we fuel the mitochondria with a mitochondrial support in the hopes that it will help slow down aging. The other aspect is immune system disorders, so autoimmune responses, as well as other types of weakening the immune response can occur, and that can cause cells to die or age earlier. And as I said, this genetic theory where this telomeres, so I think it as like end caps on the ends. So it's like if you have a shoelace and you have those little taps on the little caps on the end of the shoelace. Their job is to protect the ends of the chromosomes, but every time it divides, they get shorter and shorter and shorter. There's an enzyme called telomerase, and what this does is it lengthens telomeres, and it's only found in germ cells of embryos and absent in adult cells, but we do see in cancer cells. This is how cancer cells are able to survive. So a lot of the people are trying to study this to figure out how can we prolong our longevity. As far as chromosomes, I know in the beginning I said there were 46 chromosomes. That is correct. There are 46 chromosomes, 22 homologous pairs, which means they are equal matches. Right here is what we have a karyotype that I have laid out here. It's all of our 22 homologous chromosome pairs, and then we have a different pair, which makes up that 23 pair of sex chromosomes. Now remember, sex and gender are different. Sex in the sense of heredity in DNA, it depends if there's an XX or an XY, and that's what determines male or female. And as I said, we know my soapbox, gender-bread person. You have already heard this a million times. So what determines sex? So it is actually the sperm that determines if an embryo is going to be, or that determines if that embryo will develop ovaries or testes, because it's the sperm that carries. Sperm can carry their X or Y because it's coming from a biological male. Eggs only have XX. So when, we'll go into meiosis later, but when we're putting two cells, and we did last week, so it should have a basis of it. So ova has half of the genetic material from the biological mom, or from wherever the source is. And then the sperm has half of the biological component of wherever the sperm source is. When they come together, they're making that diploid again. But as being a biological male, they have an X and a Y. So when it splits, you have a 50-50 chance of it being boy or girl. Fun fact, I've been told that I'm a really good predictor of biological sex in a baby, as well as knowing when people are pregnant before they are. I don't know. It's been a parlor trick since I was a kid. Can't explain it. Don't ask me to do it on you, but it's really freaky. All right. With that being said, so when an X from the egg and X from a sperm come together, we have an XX, and that's a female offspring. We have an X and a Y come together, it's a male offspring. There's an SRY gene, which is the master switch for the Y chromosome. And so the SRY gene is on that Y chromosome. So that's, when it's on that Y chromosome, that gene is responsible for testes development for what we call biological maleness. So the father determines the sex of the child, which is in a nutshell. So here's this cute little babe. I love babies, sorry. Cute little baby here. So cute little baby there. Biological, so an ovate, I'm not even going to go with parents. You could be families come in all shapes, sorts, and sizes for the purposes of procreation. We have an ova and a sperm come together. They have now gone to meiosis, so they have half that, and it just shows you there. It's really 50-50, and you should be happy with whatever you get. Okay, so what can go wrong when these meiosis, when the splitting of the X and the Y, or the X and X, can occur? So we have something called non-disjunction, and what that means is that the distribution of the sex hormones, or not sex hormones, sex chromosomes to gametes, are abnormal. They're not going in the right direction. There's a syndrome called Turner syndrome, where females only receive an X chromosome, and for some reason the X or Y never came through from the sperm, and they don't develop ovaries. So these Turner syndrome females tend to have a little bit more masculinization of outward appearance. They tend to have neck webbing and shorter stance and widened nipple spaces. There are also times where more than two X chromosomes are placed with a biological female. And anytime that we have too many or too little chromosomes, we always think of an intellectually disabled person. So there's sometimes 3 X's, 4 X's. They most of the times have underdeveloped ovaries and limited fertility. And you're like, wait, they have more X chromosomes. But the more chromosomes is not always the better. And then we have the opposite, which is Klinefelter syndrome, which is males received a single Y chromosome, but somehow got two or more X chromosomes. This makes them sterile. And normally they're still, as far as intellectually normal, but sometimes can be slightly below. And what we would see there is that they still have the male characteristics, but maybe not as pronounced. So reminder about cell biology, I know we discussed a little bit before. So just before we get into mitosis, we have this thing called centrioles. Their main function is to help with cell division in animal cells. The centrioles help help the formation of spindle fibers that help separate these chromosomes out. Like if you were to take like a cotton ball, just go and divide it out. And then the nucleus, remember, that houses the DNA, and the nucleosis makes ribosomal cell units and ribosomal RNA. And remember, ribosomes help make proteins. They play a vital role in making a cell. So as I said, mitosis, cell division, replication, not meant for sexual reproduction. Mitosis is different from meiosis. Mitosis is cellular division. Meiosis is gamete production. We will get 46 chromosomes on the end, and it can occur in any cell that has the ability to replicate and is not for procreation. I think I've said that 50 million times now. It will be a test question. It will be a quiz question. It will be a word question. Okay, so mitosis, the first phase, prophase. So what ends up happening is those centrosomes shift to opposite sides of the, well, they start shifting to opposite sides of the cell. You have these things called mitotic spindles that start to form. The chromosomes will start, you'll start seeing that nuclear membrane that houses all the chromosomes that starts to break down and disappear. The nucleolus will also disappear under the microscope. So when it is not in prophase, we can see a very distinct, you'll see a purple dot with a darker purple dot in the center. For this case, we'll not see that nucleolus, and the chromosomes themselves will be apparent with two sister chromatids. They look little X's, and they will literally look like this under the microscope. Okay, in metaphase, which is our next phase, each of the chromatids face the opposite poles, so they start lining up in the center. So we have the centromeres at the edge, holding those spindle fibers. They're starting to attach to the chromosomes, and chromosomes line up in a phase. So they're equal. They're metaphase. They're in the middle of the plate. Okay. Then we have anaphase. So what happens in the anaphase is that these centromeres and spindle fibers start to pull and move to opposite ends, and they separate the sister chromatids. So now instead of the X, we have each reciprocal side of the X. And then we have telophase or telophase. And from telophase, what's happening is that you now have two daughter nuclei. So all these chromosomes have been separated. The sister chromatids then uncoil and look like chromatin again. So the nucleus will reappear, and that nuclear membrane or nuclear envelope will reappear. And that's telophase, and that's the last part. Then the last part is cytokinesis, where once that happens, we get this little contractile ring in the center. The centromeres you see slightly detached, the spindle fibers start to break down, and you have a cleavage furrow, and you have two exact replicas of the original cell. Okay, so now let's go into meiosis. So I said meiosis is the process of sexual reproduction in a sense of making gametes. So it is a unique kind of cellular division. It only happens in gonadal cells. I should not say whatever it says there. So and remember, sexual reproduction cannot take place without meiosis. So what it does is it goes from that 2N, that diploid number, to haploid. And then that haploid is present because we need to combine it again to make a diploid. I know, confusing. So some terms that we want to get familiar with as far as meiosis. So we have sister chromatids, and what these are, it's cells' DNA is replicated during interphase. Each chromosome has two sister chromatids that are identical, and they're joined together by a centromere. So that's that little X pattern with the button in between. The DNA here is coiled and condensed and makes it, so if we look at this under a microscope, you could actually see the chromosomes here. We also have what's called homologous pairs of chromosomes. That means one member of each pair is from a father or paternal side, or one is from the mother or maternal side. I should not say material. They look alike and carry the same genes that code for the same traits, but they are not exactly the same. Okay. So this is an example of here two unduplicated chromosomes, and then we have non-sister chromatids, and then sister chromatids. So see, this pair here is a sister chromatid. Okay, we have, and I already said that's non-sister chromatids. They're within the homologous pairs, but the chromatids of one chromosome are non-sister chromatids to the other chromosome. And this is important because we need non-sister chromatids to exchange DNA to what's called crossing over. And this is what makes us all so diverse as human beings. If we had no, if we didn't have non-sister chromatids and the process of crossing over did not occur, we'd all be clones and we'd have lots of genetic defects, and it would not ensure the the sustainability of the human race. Then we have the term diploid, which I've already said a few times, but there's two sets of chromosomes, one from each parent. Haploid means just half of what's being given. Okay, so in meiosis, what's happening is that for the most part, and during interphase, we're seeing meiosis, all the chromosomes are replicated, just like in mitosis. Depending on male, female, during meiosis 1, well, it's all basically the same. During meiosis 1, these homologous pairs separate from each other and reduce that number from 2n to 1n, or a diploid to haploid. This is referred to as a reduction division of meiosis. Then during meiosis 2, the sister chromatids separate and each daughter cell gets equal share of the chromosomes, and this is called also equational division of meiosis. And then in meiosis, there are similar phases, but just like in mitosis, meiosis consists of the 2 consecutive nuclear divisions, and they are referred to as meiosis 1 and meiosis 2. DNA replication only occurs once. And remember, they produce, in most part, 4 haploid daughter cells. And then each nuclear division has the same steps as prophase, metaphase, anaphase, telophase, but a little bit different. Excuse me. So here we can see an interphase. We're duplicating all the DNA material. Meiosis 1, what we're doing is we're separating out the homologous chromosomes, and then we're separating sister chromatid at meiosis 2. So meiosis 2, basically, as I said, it mirrors everything else except for the chromosomes are not replicated. There is no interphase in meiosis 2. So we can take here, there's a meiosis 2, there's prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis, and then we got the 4 daughter cells. And then as far as mitosis, remember their job is to just divide and ensures that cells in all the body have the same complement of DNA for normal growth and repair. Okay, this is all just a kind of duplicate of what I've been saying. And that is meiosis in a nutshell. We went through for each male and female how meiosis 1 and meiosis 2 look different, and you're expected to understand that and know that by now. Any questions, please reach out. So when it comes to heredity and DNA and how things are passed down, it's called genetics. Genetics is a study of this mechanism. A guy named Mendel is one who, if you ever heard of Mendelian Punnett squares, might have done those in high school biology or even 7th grade biology. He is the one who proposed these basic mechanisms when he studied pea plants and realized when he crossbreeded pea plants, there were certain traits that would come up, and he figured out if he did some predictions, he was able to predict the right thing. Remember, the genes are a recipe or blueprints for the synthesis of protein, and gene expression can be controlled by other genes. So basically everything is controlled by something, okay? Sounds very matrixy, kind of, sorry. And then a long time ago, we did something called the Human Genome Project, and this is determined that most of the genetic sequence in human DNA, and it's been really a vital tool. And the Human Genome, fun fact is the Human Genome Project, I believe it originated in Cold Spring Harbor, New York, and I used to work at that lab. Huh. 5 seconds, 7 degrees of Kevin Bacon fame somehow. No, just kidding. All right. So we already talked about how we understand haploid versus diploid, and we know that there's 22 pairs of homologous chromosomes and those are called autosomes, and they're for just about everything else in our body. And then we have sex chromosomes, and that's 1 pair. We have a karyotype, which is exactly how many diploid chromosome complements are displayed in a homologous pair, and the genome is the genetic makeup, the genetic instructions. We have a thing called gene pairs, which are alleles. And alleles are genes that occur at the same locus or same location on the chromosome, but they could be responsible for different things. And then DNA sequence can be the same or different. So we have the reason why we can sometimes have certain traits in some families and certain traits and not, because it depends if it's something is homozygous or heterozygous. Homozygous means that, or it's just a minor part, more it's a dominant versus recessive, but homozygous means that the alleles or the gene pairs are the same for the single trait that's on the chromosome. And they're the same in both chromosomes. Heterozygous means that the alleles are different. So DNA sequence is different on one chromosome than the other. And how is this important? Because of dominance versus recessive. Dominance means that if that trait is present, regardless if it's one allele or not, it will show, which means that it suppresses all the other expressions. Recessive means the opposite. So recessive means that it has to have both of those components be the same exact thing in order for it to show. So an example for dominance is it would be to have things like double jointedness. So double jointed thumbs are actually a dominant trait. I can't do it, so that's not part of me. And then recessive traits would be present only if both alleles are recessive. So something like a tight thumb ligament where the person can, where they can't do that is a recessive trait. And then we have two different things that we look at when we talk about DNA. We have genotype, which is what the actual genetic makeup of a person is. So if we did Punnett squares, we will do it in lab, we'll see exactly what's been handed to them. But the phenotype is what's been expressed. And that's what depends on what proteins are being things. So example, double jointedness. So if it's a dominant, if a dominant thing, which means that you have to have at least one allele that's dominant in there for it to show, then even though they may be big J, little j, or big J, that person will show double jointedness as their phenotype, okay? But that's different because genotype means that it's big J, or big J, little j. And we'll get into that in lab. It might not make a ton of sense right now, but we'll get that into the lab. And then as far as genetic variants, that's what intercourse is for. That's how we make sure that we aren't clones of each other, because clones means that there's a reason why families are not encouraged to procreate within the same lineage, because it leads to a lot of recessive traits that are either not compliant with life or not good to have in the gene pool get more expressed because there's not that variance available. So that independent assortment of chromosomes, meaning that when things are in meiosis are getting split into those haploid daughter cells, random choice of which one goes where. We also have crossover homologous pairs when they come together for fertile. When they come together, they cross over, and then we have some intermixing of genes there. And then we don't program exactly which sperm and which egg come together. We'll talk about that next week. Yes, we are going to talk about birds and the bees a little bit, but random fertilization, it means one egg randomly gets ovulated. We don't know which one. We don't know what genetic code it holds. And then one sperm randomly meets up. Out of the millions and millions that get ejaculated, one sperm meets up with an egg randomly. Okay. And then we have this idea called chromosome segregation and independent assortment. And what this does is it allows things to be separate versus traits. So segregation is when two alleles of one particular trait will be separated and distributed to two different daughter cells. Okay. So an example is if we have big J, little j, and allele big J will go to one daughter cell and the allele little j will go to another. We'll see that in lab. What happens is when the segregation occurs where it doesn't get distributed properly. And a common thing is Down syndrome, which is known as trisomy 21. It is chromosome 21 that accidentally gets another chromosome. And then we have independent assortment. And independent assortment means that alleles of two different traits on two different chromosomes are distributed independently of each other. This is what can make different hair, different eye colors. There's a million and one bajillion different ways to get assortment. And here's just a look at all the different ways that we can get assortment. You look at the different colors here, what comes from paternal and what comes maternal, all the different little makeups that can happen here. And then crossover homologs and gene recombination is basically that genes on the same chromosome are linked and can be passed to daughter cells in one unit. But when we have crossover occurs, it's called orchiasma, the homologous chromosomes can break, and even between linked genes, and they can recombine to become different types of chromosomes. This is really important for the success of the human race because we want recombinant chromosomes to have different expressions. That's why there's all different types of skin colors, different hair types, different personality traits, different everything, different height. That's what makes us all different. And just taking a look here of how crossing over genetic recombination occurs. So examples here, hair and eye colors. They're on the same chromosome. Yes, that's correct. Purple is from father, green is from maternal. So each one during prophase of meiosis 1, they have 2 sister chromatids. Then we have the crossing over of the chiasma occur of these of one of the chromatid segments is crossing over. They get recombined. And then when gametes are formed, we have all these different ways that we can express it. So I'm pointing, like you see, pointing a different expression. And as I said, random fertilization, there are 1 in 72, I think that's billion odds that you can recreate the same exact person. Okay. There's you, and it's going to sound so new age and yippity. There is no one else like you at all. Even your twin, you're still different in a sense. I'll get to that. I'll retake that back in a little bit, but in a sense, we're all different. Even though twins, even identical twins, 2 totally different personalities, 2 different likes, 2 different everything. So this is the reason because of random fertilization. We just can't predict. So dominant recessive inheritance, we use things called Punnett squares, and we'll go into that in a lab. And it's a great way to predict possible gene combinations. It can't be used in all genes because some genes are more than just example is hair color. Hair color is polygenetic, which means there's multiple factors coming into it, multiple expressions. But we like to look at things. Some really common things to look at is albinism. And albinism is where there's no pigment of the skin, and this is a recessive trait. So we'll take a look in a few moments about how this comes back. So we look at dominant traits as capital A and a recessive trait or a recessive allele as little a. So when we do this Punnett square, you'll see that there's all different types of ways that can come about. So taking a look right here, and it's a little bit of a smaller image. So dad has big A and little a. I believe it's little a. And mom has big A and little a. So it means they're both heterozygous, but they're not homozygous, which means there's a chance that this recessive gene can recombine during fertilization and cause a trait to be exposed. So as you can see here, because it's a recessive trait, all the dominant traits are present, whether it's big A, or big A, little a. So there's a 75% chance that this child will not have albinism, but there's a 25% chance that this kid will. And it all depends about which egg, which sperm meet up to hang out. Okay. So, and this is just probability. Predictions are just the probability of offspring inheriting. It doesn't mean that's actually has to happen. And a larger number of offspring would increase likelihood of ratios conforming to predictive values. So it's kind of like only 2 children in the family, you probably won't see much. But if you have 7 children, maybe there might be some repeats, some traits that you see. And it's all about math. I'm not going to have you do math. There's another class for that. Okay. Things like that are dominant traits that I want you guys to know about are things like a widow's peak. So in men and women, sometimes it comes out to a point, almost you think of Count Dracula. Freckles and dimples, double jointed, and the ability to roll your tongue are just a few of the dominant traits. So I guess I'm not very dominant. I have no freckles, I have no widow's peak, I have no dimples, not double jointed, and I can't roll my tongue. So I'm clearly recessive. Fun. And then there are certain disorders that they're dominant. Most disorders are dominant disorders are uncommon, because they're lethal. So death occurs before reproductive age in most dominant disorders. An example of this is Huntington's disease, which is a disease of the nervous system, and it's caused by a delayed action gene that's not activated until age 40, and that's very uncommon. Then we have recessive inheritance, which are things that there's certain diseases like Tay-Sachs disease, which is a disorder of mitochondria, yes, that typically happens in Ashkenazi Jewish. Cystic fibrosis is where the chloride ion channels are disrupted and there's accumulation of mucus in the lungs, salivary glands, and anywhere else where mucus is produced. And albinism is an example of recessive, okay? And these are more common to occur because they are not, cystic fibrosis and Tay-Sachs are deadly, but they're not lethal in the sense that you only have a very short window to live. And then we have heterozygous or carriers of the trait, which means that they can have a recessive trait, but if that dominant trait is there, or if they're heterozygous, then it doesn't mean it has to be expressed, which is why sometimes we'll see not a good example, we'll see albinism only happen 1 in every 5 generations because it depends on who gets that trait. Okay, so here are some examples of some dominant recessive genes to look out for that you should know about. So Webb digits does occur, dwarfism, Huntington's disease, normal skin pigmentation. Those are all dominant genes. And then the recessive ones I want you to know about is albinism, Tay-Sachs, and cystic fibrosis. And then we can have something called incomplete dominance, that even though we have something that's dominant that takes over and recessive, sometimes if someone's heterozygous, they may have systems, but they're not as intense. An example of this is sickle cell anemia. Sickle cell anemia is typically found in African American individuals. And what happens is that one of their hemoglobins is made abnormally and it can get stuck in places. So when someone has big S, or uppercase S, they have normal hemoglobin. Sometimes they have a incomplete dominance of sickle cell, which means that they may not show symptoms unless something causes them to have a sickle cell crisis. And then we have the little s, which means that the recessive trait is there, and it only makes this mutated hemoglobin, and they're much more susceptible to the disease. And as I said, there's also multiple things that can be multiple alleles. The most common thing here is ABO groups. So ABO, that's how we have an AB group, because there's multiple alleles that are inherited. So someone could be a type A and someone could be a type B blood who come together, and you can have a chances of having A, a B, an O, an AB. It depends on what's coming out here, okay? And then we have things that are linked to either the X chromosome or Y chromosome. We're going to really talk about mainly the X chromosome, because the Y chromosome, sorry, biological males in the class, or biological males in general, the Y chromosome only carries about 200 genes, so it's not as important. So these things that we found X-linked genes, so that's important. So if it's an X-linked dominant trait and you only have one X present and you have a Y, then guess what? You're getting that disease. Okay, so if we looked at an electron micrograph of what this XY looks like, this is the X with all the chromatid and all the chromatin coming around and genetic material, and this is the Y. Y is much shorter because it only holds a couple of 200 genes. So if the X-linked recessive alleles are always expressed in males, sorry, let's take that back. So in order for a recessive allele to be expressed in a female, they have to have both alleles on the X. But if it's a recessive thing, it's only on X and they have one Y, there's nothing to dampen that effect. Does that make sense? I hope so. So here, looking at this, for colorblindness is an X-linked trait. If the male is with normal color vision right here, and they have a Y gene, but the female is a carrier, so they're not colorblind, but they have a carrier, the odds of this coming together to make a male fetus that has her X chromosome or their X chromosome that is for colorblindness, it guarantees that that male will have colorblindness. So colorblindness is mainly from mom's issue. Then we have something called the polygene inheritance, which means the traits are a result of actions of several genes happening together. So I already said that: skin color, height, intelligence, metabolic rate. These are all multiple genes that are important. And then just remember that everyone comes in different shades. There is no such thing as the right shade of a person. It's how you're born, and it's the amount of melanin, not the amount of melanocytes, and it's a variation. We're all human. We can have different skin colors. And then environmental factors are really important on how a gene is expressed. There's certain pathogens, certain drugs, certain things in our environmental itself, things that I call endocrine disruptors, that can affect how our genes are expressed. An example that there are certain things that can be really detrimental to a developing embryo or fetus. In, I believe it's the 50s, they used to use a drug called thalidomide, and thalidomide actually affected the genetic mutations it caused genetic mutations and how things were expressed. And then other things environmental factors can express gene expression is what we put into our bodies, nutrition, how we take care of our bodies, hello naturopathic medicine. These can all affect how things are growing. And also childhood hormone defects can also lead to these abnormalities. But it's a lot of, did we get clean water? Did we get clean air? Did we move our body? Did we get good nutrition? Those are things that affect how genes are expressed. A lot of times we hear about, I don't want to get cancer, so I'm eating all these things that are good for my body. Yes, because those environmental things influence how your genes are expressed. If someone is a carrier for a gene and it can get turned on by poor diet, then we don't want to do that. Eat the rainbow. A simple way to do that. So a little different things here. This is my little soapbox here about how toxicology chemical exposures through our shampoos, things that we put on our body, the plastics that we use in our everyday, the microwaving those plastics that aren't great, leaching crap into our cells, things that affect our genes. And then we have areas that are non-traditional inheritance, which is genetic clumps that are controlled by other things. So there's things that are there's regulation that's gene expression that can happen either not from our DNA, and then mitochondria have its own little tiny bit of extra DNA, and if that is inherited from the mother, mitochondrial diseases are always inherited from the mother. That can affect things. And then as far as how we sorry, this is from me talking, not drinking water. As far as how we control gene expression, we have 3 layers. First layer is the actual protein coding genes, and this involves less than 2% of the cell's DNA. It's the blueprint. The second layer is what we call small non-coding RNA, and this is about 80% of our genetic genome. So only 2% of our genetic genome is actually responsible for blueprint. The rest is the small non-coding RNA, and they have a part that we won't get into because you guys are not molecular biology people. And then we have a third layer, which is epigenetics. So it's just stored in proteins and chemical groups that are bound to our DNA. Epigenetics is a big field that's going on right now because we're seeing what our parents have carried on and their grandparents are carrying on, what has been tagged to our DNA or bound to our DNA for generations, and this can influence our health. Epigenetic marks such as genomic imprinting. We typically have genetic imprinting that occurs to certain genes that are tagged with what's called methyl groups, and this is essential for normal development. Methyl groups, you might have heard things about methylated vitamins. It's the same idea. And as I said, epigenetics is a hot topic. It's something I talk about a lot in practice. I'm not going to go into this too much because you guys can leave that all. And remember, mitochondrial DNA is inherited from the mother only. Mitochondria actually have 37 of its own genes. And mitochondria are transmitted to embryo by the cytoplasm of the egg only. So errors in mitochondrial DNA are actually linked to very rare disorders. But normally what we see is these weird oxidative phosphorylation or cellular respiration issues. It's a lot of times certain seizure diseases are attributed to mitochondrial diseases. That's when ketogenic diet is used for treatment. And then Alzheimer's and Parkinson's disease, we don't have enough evidence linked to it. Well, we have a lot of evidence, but I think it might be a mitochondrial error rather than anything else. And we could always, we can look at your genome now and tell you what genes you have for the most part. You've heard of 23andMe, the genetic genie, etc. So we could do genetic screening and genetic counseling to help parents out. So example is for someone who's Ashkenazi Jewish descent. I mean, they're from Eastern European and they have Jewish lineage. They're, for some reason, have about 20 different genes that are more common in that population. And what they do, and I didn't make this name up, but it's called the Jewish Panel, that is run on pregnant women who are from Ashkenazi Jewish descent to make sure that they don't have a lot of these diseases. Other things that when a newborn is born, we're doing genetic screening already from the start. Their first few hours they're born, they get a heel poke. And what that heel poke does is it gets blood out. We send it out to a lab and we screen for things like PKU and congenital hypothyroidism, stuff that we can nip in the tushy right away. The fun thing is that a lot of these diseases don't show up until about the 7th to 10th day of life. So we actually do 2 newborn screenings. We do it on day 1, and then we do it 7 to 10 days later. And then examples that for screening adults. So examples are the BRCA genes that we talked about with breast cancer. We can do genetic screen for that. If someone has a history of Huntington's disease, we could test that. If women who are 35 or older, they typically test the fetus for trisomy 21 because for some reason 35 is the magic number when trisomy becomes more potent. And that could be because that telomere shortening and aging. And then how do we figure out, or what's a good way to figure out what came from where? We do pedigrees, just like doggies. If you ever see dogs get their pedigrees done for proof of purebred or not. So we could do these pedigrees that kind of take a look at generations and generations and generations and see what came from who. So this is a look at someone who has congenital dwarfism, who from here has it and what are the likelihood of them happening again. As I said, there's blood tests that can be done. So Tay-Sachs and cystic fibrosis are things that we can do as a test. There are blood tests that can also look for sickle cell genes if there's issues with that. When we want to prevent things from it becoming once they're born, there are two things that we can do to test a fetus to know if there's a genetic risk of them for them to have a disorder. Sometimes this can influence how that fetus will develop. Sometimes it influences the mother's choice about what they want to do. I'm not getting into the politics or any kind of moral things about that. But we have two ways to do this. We have amniotic fluid testing, which is called amniocentesis, where they take a little bit of the amniotic fluid through out from the womb. At week 14, they test the cells in there and test for genetic abnormalities. We also have chorionic villus sampling, which they take we'll learn about placental formation soon, but they take a little bit of the tissue that attaches to the in the placenta, and they test it at 8 to 10 weeks to karyotype for genetic abnormalities, and this can be done earlier. So this is taking a look at each one. They both require testing. There are certain things now where they can do what's called a quad screen, and that can be done as early as 4 to 5 weeks of a person who's considered a geriatric pregnancy, which is nowhere near geriatric. I hate that term. 35 and over is considered a geriatric pregnancy, which heck no. And we also can now realize that we can use genes to help alleviate or cure diseases through therapies or manipulating those genes. So with the genetic engineering, we're hoping that we can replace genetic defects or even rebuild tissues, which they've done already. They've made a heart, they've made a couple of things inside the lab. There's some controversy around this about where those cells are coming from, religious aspects, societal aspects. That is a whole can of worms that I'm not going to open. I hope this was informative. I know genetics is really tricky. I don't want you to concentrate so much on the nitty gritties. I want you to get the main concepts. The main concepts I want you to understand are the different terms to understand how we get genetic variants or how we all don't come out like clones in the end, and the difference between mitosis and meiosis, including the different phases that occur. Thank you so much, and I hope you guys have a wonderful week. Bye-bye.